Okay. Kelvin. We'll zoom. Oh, is this as far as it goes? Oh, Jesus. Okay. So carbon cycle, sorry, not the carbon cycle, the Kelvin cycle, which is fixating CO2 and bringing it into the plant. And then I'm also going to hopefully finish photorespiration. If not, it will just kind of bleed in tomorrow. But basically life from air. Mouse, where are you? Okay, so the Kelvin cycle, we got to remember, what does it mean to be a plant? And it means that you got to make your own food because you are an autotroph, but you got to be able to store energy, in this case ATP, and as well as electron carriers from light reactions. We got to convert ATP into a more stable form because ATP is unstable. You only have a short amount or a small amount of it before it's completely used up. You got to be able to move it throughout a plant, and these organisms can be huge. Think about redwood trees over 100 meters tall. Also, you got to save it for a rainy day because you may not always get that sunlight. Okay, so this is what we're going to talk about today is the glucose part. But again, please know photosynthesis, the chemical equation. You're going to take in 6 molecules of CO2, 6 molecules of water, a little bit of light, make your glucose and 6 molecules of oxygen. So yesterday we talked about water entering in with the photosystems and being split and donating electrons to the reaction center. And now today we're going to focus in on carbon dioxide. So to build carbs, you need some energy and you need some electrons. And we have generated that energy, again, with the photosystems through cyclic phosphorylation. And we've also generated some electron carriers, NADP plus, to form NADPH. So we got the energy and some electrons. And so what can we do now? We can build stuff. And so the word photosynthesis means light put together. So with light and these materials, as well as some CO2, we can put together and build an organic molecule that the plants can use, store, and save for a rainy day. So remember, in order to make C6H12O6, these hydrogens usually come from the water, and then the carbon and the oxygen are going to come from the CO2. So what raw materials are available? Well, today we're going to focus in on that carbon dioxide and 6 molecules of it to be exact to form that glucose molecule. So what's being reduced? We're going to reduce CO2. That means gain electrons. So you can see that electrons being transferred to the glucose molecule, and this is being oxidized. So CO2 has very little chemical energy by itself, and so we need to convert it to C6H12O6. Again, synthesis is an endergonic process. You have to put in a lot of energy, and in this case, I think it's going to take about 18 ATP. So we're going to reduce CO2 to C6H12O6 in small little steps, and so you're going to see that with the Kelvin cycle, how there's multiple little steps as it goes through, as we add on another carbon and rearrange some atoms. You're going to find that the Kelvin cycle is very similar to the reverse of glycolysis.
Did we decide that the oxygen that it gives up, that comes from the water?
Yes. So we talked about the light reactions yesterday, how electron carriers and ATP are made, and now we're going to use it into the Kelvin cycle, take in CO2, and form our carbs. So this, I should say, the Kelvin cycle takes place in the stroma of the chloroplast, where here it was within the thylakoid and the thylakoid membrane. Okay, so we're converting inorganic carbon into an organic molecule. And so this is the Kelvin cycle, cycles of reactions that allow carbon fixation, discovered by a guy named Melvin Kelvin. You can't make that up. And what he did was he kind of first noticed this first intermediate molecule and then how it got changed through a series of reactions. And so they named the cycle after him. So a key step is taking CO2 from the atmosphere, this inorganic form, and bringing it into the plant. And how plants do that is they need an enzyme that's called ribisco. Okay, so ribisco stands for ribulose bisphosphate carboxylase oxygenase. You don't need to know that, you just need to know ribisco. So you can kind of see how they formed the word ribisco, ribulose, bis, and then carboxyl ribisco. It's a very large enzyme found in the stroma of chloroplasts, and what it does is it takes a molecule called RUBP, which stands for ribulose 1,5-bisphosphate, so we'll just call it RUBP, and which is, so I just said, it's a 5-carbon molecule, and then this is 1 carbon, and we're going to take it and we're going to bond it to each other using this enzyme, ribisco. So RUBP, 5-carbon sugar, binds with CO2 to form an intermediate molecule that immediately splits into PGA, which is a molecule that you should be familiar with from glycolysis. That was one that you had to memorize. Okay. So ribisco. What?
That was PEG.
Yeah.
That was like BPG.
Oh, there was like 10 intermediate molecules. There's probably one you didn't have to memorize because I probably gave it to you, but it's in there. All right, so this enzyme ribisco fixes carbon from the air. It is probably the most important enzyme in the world because from it, this is where life basically comes from, because like you use sugar. Every organism needs to acquire energy, and we take it from CO2 and convert it into this organic form. It is by far the most abundant enzyme in the world. So here's the overview of the Calvin cycle. You have your 1 carbon CO2, okay, coming into the Calvin cycle. Here's RUBP, a 5-carbon molecule, and they will bond together using this enzyme ribisco to form an intermediate 6-carbon molecule that's unstable and will immediately break down into PGA. Then we're going to take some energy and rearrange and add some phosphates and do some things with it, drop off some electrons, but eventually you're going to get to G3P, and then one of the molecules of G3P gets set aside, and the cycle gets rearranged. We use some more energy. We go from 3 to 5, and it goes again, and then a G3P molecule gets set aside. And then these G3P molecules are the things that actually get bonded together to form your glucose, which can be modified and bonded to other glucose molecules to form starch, sucrose, cellulose, and much more. So remember glycolysis, right? Where we start with glucose, and glycolysis means to split sugar, and it splits into eventually 2 molecules of pyruvate. Well, photosynthesis is the reverse process of that, but we're not going to start with pyruvate. We're going to start with the PGA, basically. So 2G3P and beyond. G3P, if you don't remember, stands for glyceraldehyde 3P. End product of the Calvin cycle. It's very energy rich. A lot of times we call it C3 photosynthesis, and we'll talk about that because...
Staff members, please limit movement between your classrooms or within your classroom. Students may move between classes though when the bell rings. Thank you.
So G3P is a very important intermediate molecule because from it you can make glucose, but then it can be rearranged and can form lipids, amino acids, nucleic acids, which is the 4 macromolecules of life. So, and it all comes from G3P. So that's kind of cool. Okay. So glucose is not produced directly by the cycle. Remember when I said that G3P gets like set off to the side and the cycle goes another round and another one gets set off aside? It's produced indirectly by the cycle because it's trying to get G3P, set one aside, continue on, turn into ribulose, take in another CO2 and go around again. So here's the net equation for the Calvin cycle. 6 molecules of CO2. You're going to use 18 ATP in the process of the Kelvin cycle, as well as 12 electron carriers plus water indirectly from the photosystems, and you're going to form 2 G3P molecules because G3P has 3 carbons in it, so you have 2 of them, there's 6. 16 phosphates, 18 ADP, because these guys get oxidized, and then 12 NADP plus.
Why does the phosphate always have a little eye on it?
It means inorganic. If the P was bonded to a carbon and an oxygen, then you could, and even a little hydrogen, then you can call it organic. But it's just phosphate by itself. Okay, now in order to form one molecule of glucose, the cycle has to go through 6 times. You're going to take in 6 carbon dioxide, but you can only take in once, as you go around. So this has to happen 6 times. 2 G3P molecules will be eventually produced. 6 molecules of RUBP will be regenerated to take in some more carbon dioxide. So just understand, in reality, it has to go through 6 times. Okay? And realize that when it goes through 6 times, those carbon dioxide molecules that got taken in do not end up in the same molecule of glucose. It's kind of like an assembly line. I don't know if that makes any sense. No, that didn't. Okay. I can't even describe it. It's like it had to start somewhere, and
Like the first 6 went to the first molecule. And then
possibly, yeah. And then it just kept evolving backwards until this is where we're at right now. But yeah. So
it's basically just not all of them go to the glucose.
Yeah, right. But they do, end up in a glucose molecule, just not the same one. Now, the accountant is super complicated. Depending on how you look at it, how does your book simplify? There's different diagrams of the Kelvin cycle, because maybe they just do it, 2 times and you get 3. Maybe they say 3 CO2 enter in at one time. That's not true. It's just 1. So I don't know. With 3 turns, you get 1 G3P, but with 6 turns, you get 2 G3P that can be used to make glucose. So it's just depends on how you look at it. But eventually, it is 18 ATP and 12 NADH for 1 glucose molecule. If there is any ATP left over, it just gets used by the cell elsewhere, to move things. This is just saying for sure 18, and then anything left over goes elsewhere. Okay, let's dive into the Kelvin cycle. So you can break the Kelvin cycle down into 3 phases. The carbon fixation, which means that we take in a carbon dioxide molecule, bind it to ribulose, and eventually get to PGA. And then the second stage, after you get PGA, you're going to reduce it to eventually 12 G3P molecules, which is the reverse process of glycolysis. 2 get set aside, 10 move on and get rearranged into RUBP. So, yeah, carbon fixation, reduction, regeneration. So I think you need to know those terms, because on your Kelvin cycle, I think it's broken up like a little pie chart, or it's very similar to it. And you have to write reduction, regeneration, fixation. So notice this cycle, they have 3 CO2 coming in. So again, you just got to look at the diagram and figure out what are they starting with. So CO2 comes in, binds to RUBP. Okay, so this is a 5 carbon molecule. Here's my 1. It forms an intermediate molecule that is unstable and breaks down into PGA. And there's 6 of them, like this. Okay. Do you guys want to count the carbons? Do you think it would help if we kept track of the carbons?
Sure.
Okay. So these little red circles represent carbons. And you have 3 of them, so 3 times 1, 2, 3, 4, 5. Okay, so we have 15 carbons here. And then 3 are coming in, so 15 plus 3, 18. And that makes sense because here's the 3 times 6. So now I have 18 carbon. I should just put a plus 3 carbon, right? Okay, so then I guess we can also keep track of the phosphates if we really wanted to, but we have 6 6 P's over here. Yeah, we'll just do that. Okay, so now ATP comes in, and we're going to reduce PGA to get to G3P. So here's ATP. It's going to lose a phosphate, but there's 6 of them, so it's going to lose 6 phosphates to it, as well as some electrons. And oh, they don't show that intermediate molecule. It's in between. Crumbs. Well, anyways, okay, so we still have 18 carbon here. We still have 18 carbon here. 6 phosphates, which is the same, so we're okay, but we should have gotten 6 more. Maybe these guys just float and do something else. Okay, so now we're going to set 1 G3P aside and continue on. So now instead of having 6 G3P, we have 5, but we now have 5 times 3, right? Still with me?
Because you had 3 other.
Yep, because 3 went over here. So this is 3 carbon. So 18 minus 3 is 15. Does that work?
Oh, 15 carbon.
Okay. Then we're in the regeneration phase of taking G3P and forming it back into RUDP. So we're going to use some more energy, and then these 3 phosphates get added to that end. So we still have 15 carbon, and then the rounds goes again. Yeah. Okay, so this is a light independent reaction, which means light isn't needed for the Kelvin cycle to continue. But to get the ATP and the electron carriers, we need that from the light dependent reactions. Light promotes the transport of all the intermediate molecules. Light promotes influx of magnesium to further activate rubisco. So, I mean, it is dependent on light, but it can take place at night, and you're going to see that with some plants. So in summary, we have the light reactions. We're making ATP, electron carriers, water is split, oxygen is a byproduct. In the Kelvin cycle, we're taking in CO2 from the atmosphere. We're producing G3P, which will eventually turn into glucose. We regenerate ADP and NADP+, and these guys go back to the light-dependent reactions. So this little flow chart, I think, does a really nice job. Hey, we make the energy, the electron carrier. They're used in the Kelvin cycle. They come back to pick up more electrons and to make ATP. That's kind of cool. So again, remember what comes out of the light reactions: ATP, NADPH, and oxygen. This is where water enters. And then for the Kelvin cycle, make sure you know what goes in the Kelvin cycle and what comes out. CO2 comes in. We form NADP plus ADP and sugars. So then when you put it all together, this is what you get. So it's kind of like the circle of life, right? Because we discussed, what?
You go back up really quick.
Oh yeah, sure.
What's that? Oh, that's what it wasn't labeled on ours.
Okay. So it's like the circle of life, because we just finished cellular respiration, where you take glucose and oxygen, which is a combustion reaction, and you generate CO2 and water as a byproduct. And then photosynthesis is the reverse of that, where you take in CO2 and water with a little bit of light, and you get sugar and oxygen. Plants are the only ones, well, I shouldn't say only, but go through photosynthesis. Cellular respiration does happen in plants. They do have mitochondria. They can burn their glucose and use it, but animals too. So a little summary, I think on your guided notes we have some questions about like where did the CO2 come from and where did it go, right? So where did the CO2 come from?
The air.
The air, the atmosphere, okay? Where did the CO2 go?
Goes into the glucose.
Yep, G3P. So it enters the Kelvin cycle to become glucose. Where did the water come from? The outside, the soil, you could say the xylem of the plants. Where did the water go, or what happened to it? It's made into oxygen. It's split. Awesome. Okay, where does the light energy... Oh, I just said the answer. Never mind. Where did the energy come from to do this? The sun. Okay. What's the energy used for?
To make the NAD plus.
To make the NADPH and the ATP, because light-dependent reactions, that's what's being made. So they capture the light energy, they excite those electrons, they create ATP, and those electrons get picked up by NADP+.
Is that in the life-dependent reactions?
Yep. Okay. What will the C6H12O6 be used for? Lots of answers for this, but what's glucose going to be used for? Energy, food storage. You know, you can make all the macromolecules from it. So basically, the work of the plant's life. Where did the oxygen come from?
Breathing in. Oh, from the water.
What?
The water.
Yeah, the water. So from the splitting of water, and then where does it go?
Just 6 oxygen.
Into the air. It's going to travel through the stomata. If you remember that, I don't know if you do, but it goes back to the air, or it goes to the air. All right. What else is involved? And it's not listed in this equation at all.
The NAD+.
Yep. Which is reduced into NADPH, and then what else? ATP. Yep.
Wait, so in the end it's given off as NADPH and then ATP. It's not the NADP+. Like in the end of all of it.
Say that again. Wait.
Starts with NAD.
So it starts with NAD+.
Yep. And then it picks up the electrons
In the light-dependent.
in the light dependent, and then to make the glucose, it becomes NADPH to turn into NADP+, and then it goes back. So it's actually on both sides of the equation, depending on how you look at it. That's why they're not in the equation.
Also, H+ is like protons.
Yes. Okay, so on a global scale, photosynthesis is probably the most important process for the continuation of life on Earth, because we get, like you take this organic carbon, you turn it into an organic form that you can use. So it captures over 121 billion tons of carbon dioxide from the air, makes over 160 billion tons of carbohydrates. Heterotrophs, they're dependent on plants. You can trace your food back to a plant and ultimately to the sun. So in a poetic perspective, all the solid material of every plant was built by sunlight out of thin air. That's kind of cool. All the solid material of every animal was built from plant material. So really, we're just particles of air woven together by strands of light. And I think Mr. Rourke has a poster like that, or a quote on his wall. So when you have chemistry, I think there's one. Okay. All right, so remember what plants need. They need, obviously, light, water, and CO2, and where it happens. And now I want you to think just briefly about the structures that plants have evolved to get these materials. With roots, they have tiny little root hairs to increase surface area to get, to take in water. They have xylem and phloem to move sugars and water up and down the plant. Their leaves are usually, to increase surface area to get as much sunlight as they can. The stomata are underneath the leaf to release gases, water vapor, and CO2. So when we get to plant structure and adaptation, second semester, it's actually really cool how form follows function. Okay, the last section I want to talk about is photorespiration. The enzyme Rubisco, it can do what it needs to do, but it's not the best enzyme out there. And scientists can't figure out why plants still use Rubisco or haven't evolved a better way to fixate carbon dioxide into the plant. Okay, so sometimes evolution doesn't always result in all optimum solutions. Okay, like I said, this enzyme, it's faulty. It's not 100% efficient, and the reason for that is because oxygen can be bonded to the same place where CO2 comes in, and as a result, you can't go through the Calvin cycle. So we call this photorespiration. Eventually, what photorespiration does is it's going to reduce the yield that a plant can produce. Carboxylation and oxidation of RUBP can catalyze at the same active site for Rubisco. That means carbon dioxide and oxygen compete for the same spot to bind to that enzyme. So we call this competition inhibition. If you remember from enzymes, I think that was like chapter.
Inhibit means it allows them to do it.
No, inhibit means it prevents. So plants want CO2 to bind to Rubisco, okay? But oxygen competes for the same spot. And oxygen's being a byproduct in the plant, so sometimes it doesn't leave the plant, stays in the plant, and it binds to this enzyme, and it just messes up the Kelvin cycle. They've looked at alter in temperature increases, what happens to the loss of reducing yield. So at 25 degrees Celsius, carboxylation reaction occurs 4 times more than oxidation, which is good. Carboxylation means we're fixing carbon. 20% of photosynthetically fixed carbon is lost to photorespiration. So as temperature increases, the loss continues to rise. So like past 25 degrees Celsius, this is where it gets hot and arid and the stomatas close because they don't want to lose water vapor and wilt. So closing cuts off the supply of CO2 entering the cell because we're closing the stomata so CO2 can't get in. And as a result, the oxygen that's being produced in the Kelvin cycle, the light reactions is what I meant to say, is still inside the plant and has nowhere to go. So then it binds to rubisco, and then thus decreasing your glucose output. So low CO2 and high oxygen favors photorespiration that occurs inside the plant. So there are some ways to control water loss from the leaves. So on those hot and dry days, plants will close their stomatas. Stomatas is the opening, and then what makes the stomata are these 2 guard cells. And it's water pressure that allows it to open and close. So when you have lots of water, they get plumped and they can't touch each other in the middle. So then you get that opening. And then when it loses water, it wilts and shrinks and it closes. Okay. So this is an adaptation to living on land, but it does create some problems because they're like, oh my gosh, we're losing water. I got an idea. Let's just close this. And then we'll just further prevent water vapor from escaping or coming in. Okay. So when stomatas close, you get that oxygen built up inside the leaf from those light reactions. Your CO2 gets depleted because you're not taking in any more CO2 to enter the Kelvin cycle. So the inefficiency of rubisco. Like I said, this enzyme doesn't work that great. Usually the CO2 will bind to it. That's the optimal substrate. You can make sugars. But when you increase those oxygen levels and decrease CO2, now oxygen will bind to it. And BP becomes oxidized and sugars start to break down. So instead of making glucose, you get glycolysis basically. So this is why we call it photosynthesis. We make sugars and then we break it down photorespiration, kind of like cellular respiration.
I was thinking about this yesterday.
Mhm.
And if they say like, isn't there like saying that like the carbon emissions or whatever, there's like a theory that they're getting too high because of like cars and stuff like that. Why don't we just plant a ton of trees?
Oh, yeah, it should. It should. When you think about the percentage of CO2 in our atmosphere, it's 0.03.
That's yeah.
So it kind of depends on the data you're looking at, because a lot of times they're biased.
Yeah, no, I know, but like even if that was the case, then I would be like, okay, let's
Well,
just why don't they get out there,
if I wasn't
Get
being
their work.
recorded, I'd say something else.
get their work boots on, and start planting
Whisper
trees.
it to us.
Well, it's just that Earth has gone through warming periods
Yeah.
before, and I think that's what we're going through right now. Is the warming. Like for the next couple decades, it's going to be warm, and then we're going to go through another cool. It's just the way, yeah.
Well, I
And
mean,
everyone's
and plus the fact that like they say that all the recycling we did and stuff in the last 10 years was just washed away when the volcano erupted. It's like, isn't that your sign that like we don't really have that much control over it?
yeah. We may not have control, but we can do what we can to, assist. Even. All right. So the Kelvin cycle occurs when CO2 is abundant. Yay! But when the Kelvin cycle doesn't have CO2 and has high oxygen levels, this is what happens. Oxygen comes in, binds to RUBP through the enzyme rubisco, right? And now this sugar molecule, instead of being, intermediate 6 carbon, basically just gets broken down immediately into smaller molecules. So it goes to the mitochondria, and we use it for respiration, but we don't make any ATP really in the process. Wow, why did I skip? Gosh, why does that happen? Okay, so there are some ways to prevent or try to reduce photorespiration. So one way is to be a C3 plant, which means you fix carbon using the Calvin cycle, right? CO2 enters the Calvin cycle in the mesophyll cell. Okay, but there are some alternative ways. So like most plants are C3, but there are some plants out there that that live in those hot, arid conditions where they can become different plants. So we call them C4 plants or CAM4 plants. So alternative mechanisms to prevent in a C3 plant. I'm kind of jumping the gun here. Add CO2 to PEP to form a 4-carbon molecule. In this case, we'll use the enzyme PEP carboxylase. 2 advantages: greater affinity for CO2 than Rubisco, but we don't really have that oxidized activity, so it's not as easy. It's like trading apples for this. It's like bringing a lemon to a knife fight. Okay, right? Okay, maybe not.
I never bring a knife to a gunfight. I've never bring a lemon to a knife.
It's like it's gonna hurt, but doesn't. Yeah. Okay.
Spray it in your eye.
That's what I mean. Okay. Oh, I shouldn't. Okay. Anyway, so now instead of being the 5-carbon molecule to a 6, we get this like 4-carbon molecule intermediate that gets modified and eventually decarboxylizes. So that's one way that C3 plants can overcome photorespiration, but it's respiration is still going to occur. Okay, other ways is to separate the carbon fixation from the Calvin cycle. So instead of having CO2 come into the Calvin cycle directly, we can physically separate carbon fixation from the Calvin cycle. And this will reduce competition for oxygen, CO2, Rubisco. So different cells fix carbon versus where the Calvin cycle occurs. You store the carbon in 4-carbon molecules, and then you have an enzyme that kind of shuttles it over. In this case, PEP carboxylase. Different leaf structure. That's the main thing. The leaf is set up differently. Now, in CAM plants, which are your oh my gosh, I can't think. Well, I was going to say cactus, but there's another word for it.
Cacti.
No.
Yes.
Nope, I can't think of it. Okay. Anyways, CAM plants, so like cacti, things that can like aloe vera. Yes, thank you, Danica. I was like, I wanted to say like flatulence. I'm like, that's not it.
I have
That's
a cactus
completely.
in my room, and his name is Henry, and he's dying.
How
I
can
don't
you?
know how much of it is water.
Well, yeah.
Water and
Okay, so in CAM plants, their thing is they're going to separate carbon fixation by time of day. So they'll fix carbon at night, not during the day. C4 plants actually have a separate compartment that allows CO2 to come in. So CAM plants, you fix carbon during the night, you store carbon in 4 carbon compounds, and then during the day you perform the Calvin cycle, because then you'll close the stomatas. Oxygen can't, enter, and wow. Okay, so C4 plants capture CO2 in one cell that's adjacent to the Calvin cycle. So it's spatial or physically separating the two processes. CAM plants, temporal solution to photorespiration. Capture CO2 at night, decarboxylate it during the day. But it's in the same cell. I thought I had a picture. Oh, I do, coming up. Okay. So C4 plants evolved to minimize photorespiration. So some examples of C4 plants: corn, sugarcane, sorghum, grasses. And they'll fix carbon in mesophyll cells. So I think on your guided notes you were like, what is the function of a mesophyll cell, right? You'd probably want to say in C4 plants it fixes carbon. All right, so as we bring in carbon in the mesophyll cells, eventually it's going to produce pyruvates and some carbon dioxide, and it's going to get shuttled and eventually converted back to PEP. So it does have a cost. You're going to burn a little bit of ATP doing this. But here's the anatomy for it. C3, we just have these open spaces and stomatas that allow things in and out. But in a C4 plant that's spatially dividing it, this is where carbon comes into the mesophyll cell, and then it will enter into the bundle sheath cell for the Calvin cycle. And then what the sugars hook up to the vascular tissue, the phloem, that's what I wanted to say, the phloem, and then oxygen will be released elsewhere. But. So it physically separates, can't say that enough, the processes.
Whereas like C3 doesn't.
C3, it's all in one place. Like it all happens just in one place. But in C4, you zoom in, you see it's really organized. Here's your vascular tissue. There's the bundle sheath cell, and then there's your mesophyll cell. So again, comparative anatomy, they're physically separated. Here's C3, again, another open diagram. You can see that we have the mesophyll cells up here. We have some spongy mesophyll, but here it's just more organized, centered around that bundle sheath. Two different compartments. Okay, now for CAM, it's in the same place, but different times of the day. So during the day, they close the stomata. At night, they open them. At night, when they open them, they're taking in CO2. In the daytime, they'll take that CO2 that they stored and bring it over to the Calvin cycle. So succulents, there's the word. Some cacti and even pineapple are considered CAM plants. So they're splitting it into night and day. And I just said this. It's just more words saying the same thing. Repetition, right? Do you guys find that repetition does it make it more confusing or more clear?
More clear.
Does it?
After about 40 tries.
Yeah.
40, okay.
Just have 40 slides of the same thing. I'll eventually get it.
Okay, so to compare C4 and CAM plants, remember C4, different compartments or different places inside the leaf, and CAM, same place but different times of the day. So here we're fixing CO2 in a different place, and then it gets shipped over to the Calvin cycle. In CAM, it's in one place, but they darkened it to show nighttime for carbon fixation and then light to do the Calvin cycle.
Wait, so pineapple grows out of the ground like that?
Not out of the
You know, to be honest, I've never heard of the pineapple juice. I never really knew how they do.
Okay, so why do we have this C3 problem? They think that it has something to do with Earth's early atmosphere composition. How Rubisco evolved in high CO2 atmosphere levels, wasn't strong enough to select the two. Today it makes a difference because when you look at composition, 21% oxygen. There's more oxygen today than there was back then. Back then there was hardly any oxygen in our atmosphere. But because of volcanoes and early photosynthetic creatures, cyanobacteria, 21% oxygen, 0.03 carbon dioxide. So photorespiration can generate about 50% of the carbon fixed. So that's a lot, of glucose that's not being made.
So is the atmosphere ever going to run out of CO2? Will people just keep breathing it out?
I don't think it could ever run out. I mean, I think
What if all the people that died? There was no people to breathe it?
there was no people. You still have animals.
What if all the animals died?
You still have carbon dioxide that's in the oceans.
So after a while,
It might, but carbon dioxide, right, or like most of the CO2 is found in the oceans. And because ocean temperatures are rising, it's kind of like when pop gets flat, the CO2 escapes. That's what's happening right now. Okay. So quiz tomorrow on just the photosystems. Okay. Diagrams in your book. I don't know what figure it is.